Do you know anyone that has been affected by breast cancer or type 2 diabetes? Perhaps you have even had firsthand experience with one of these diseases. Today, millions of people suffer from breast cancer and type 2 diabetes worldwide. Hello, my name is Caroline Nelson. I am a junior Marquis Star Fellow and Stamp Scholar majoring in biology. The research presented here was conducted as part of my continuing work with Dr. Patel in the Department of Biology at the University of North Carolina at Greensboro. 1 in every 8 women will develop an invasive breast cancer during their life. Type 2 diabetes is the 7th leading cause of mortality worldwide. Finding an effective treatment for both of these diseases could save millions of lives. The majority of breast cancers are estrogen receptor positive or ER positive, and their proliferation is fueled by the hormone estrogen. Tamoxifen, a selective estrogen receptor modulator, has long been a popular drug used in the treatment of breast cancer. However, many patients eventually develop a resistance to it. Insulin-resistant type 2 diabetes is characterized by an imbalance in the formation of lipid, lipogenesis, and the degradation, lipolysis. The flavonoid naringenin is present in many fruits, but predominantly in grapefruit. It has been found to affect the metabolism of lipid by disrupting the accumulation of lipid droplets within adipocytes. Naringenin also promotes cell death or apoptosis, and reduces proliferation in breast cancers that are both susceptible and resistant to the estrogen receptor antagonist tamoxifen. The methods through which naringenin works are unknown. Our experiments hypothesize that the flavonoid shifts the profile of the cell's metabolism to be more anaerobic. We also hypothesize that naringenin's effects in breast cancer cells can be attributed to an interaction between the flavonoid and the estrogen receptor alpha as well as the estrogen-related receptor, both of which play major roles in the metabolism of breast cancer cells. MCF7 estrogen-positive breast cancer cells were cultured in the presence of tamoxifen until confluent. The cells were then treated with MG132, a proteasome inhibitor, naringenin, or both. Insulin-resistant 3T3L1 adipocytes were also cultured in the presence of DMSO, a vehicle control, and naringenin. Next, the proteins derived from the cell lysates were separated into a 10% polyacrylamide gel. The relative abundance of each protein was then determined and quantified using Western blot techniques. As seen in Figure 1, the insulin-regulated glucose transporter type 4, GLUT4, plays a critical role in the transport of glucose into the cell. The transporter is regulated by the hormone insulin, and when activated, allows for the diffusion of glucose into the cell. Shown in graphs 1 and 2, the relative abundance of the protein decreases in the presence of naringenin in both breast cancer cells and adipocytes. Shown in Figure 2, the estrogen receptor alpha is a transcription factor that binds to the promoter of several oncogenes that are active in breast cancer. The estrogen-related receptor alpha is an orphan nuclear receptor whose presence has been correlated with the proliferation of both estrogen-positive and estrogen-negative cancers. Our results, summarized in Graph 3, reveal that the presence of naringenin decreases the concentration of both estrogen receptors. Our findings suggest that the flavonoid naringenin produces its effects in breast cancer cells and insulin-resistant adipocytes through the regulation of several proteins crucial to the formation of the diseases. These conclusions, as well as that of previous studies, indicate that naringenin has the potential to be a powerful therapeutic agent. Moving forward, our experiments will focus on identifying additional metabolic proteins to further elucidate the mechanisms of naringenin. Thank you.